It's Aksimentiev, physics department here, and I will tell you a little bit about one particular project, which will also showcase the repertoire of tools that we have for modeling the interface between biological and nanotechnological system. Again, please feel free to ask questions as I go, okay? All right. So, you know, when it comes to engineering microscopic systems, we often think of computer modeling as yet another tool, right? So, airplane were designed and previously using air tunnels, right? Where you put a model and look at the lift. But now you can do basically same kind of modeling using, same kind of experiments using computational modeling. Same thing goes to designing cars, and pretty much everything that is in macro world that is successful utilizes computer tools to some degrees, right? So I guess we're all on the same page here. Now, but if we go to nanoscale devices, situation becomes a little bit more different. Well, because they're really small, you know, there's a way to image some of the devices using TM or AFM or so on. But when you go to a scale of a few nanometers, you really don't have a choice. Yeah, you don't have a good way to look at things, especially when they are at the interface of bio and nano, which usually implies water. So in situ TM, you know, has been developed, but it's not yet at the stage where it can reveal the microscopic processes, for example, action of an enzyme or assembly of a protein at the interface of a silicon chip and a protein. So here's some figures that shows you the scales that bio things happen. So, you know, well, we are a bio thing, right? Our scale is 2 meters, and then it goes down from there. So human hair is about 50 micrometers, yeast is about 4 microns, HIV virus about 100 nanometers, a protein 10 nanometers. An ion, 3 angstroms. A hydrogen atom, 1 angstrom. There's nothing less than that we care about. Okay, so this is a very brief intro to the methods that we use. We use all-atom molecular dynamics, which is a kind of a computational microscope that allows us to image microscopic processes that take place in various systems. So typically we have a big computer like Blue Waters, a system that is available here at Illinois, but Illinois is not the only place. There are quite a few of those. We just happen to be one of the largest ones. And what we do is we build atom by atom microscopic models of systems of interest, and then we animate them by solving equations of motion, just classical equations of motions, F equals ma, many times, and at the end of the day we obtain what is called a trajectory, which is basically describes how atoms' position change in time. So this is all-atom molecular dynamics, typically involves water, ions, and everything is moving. All right, so I'm going to tell you today about one particular project that we have been working for some time. And but before I do that, let me remind you that a biological cell, you can think of it, it's like a machine, right? So you have a DNA molecule that contains all instructions that are necessary to manufacture everything, pretty much. RNA is a working copy, and proteins are the things that do the job, right? And all of them are linear polymers, by the way, right? So DNA is a linear polymer, RNA is a linear polymer, protein, if you unfold it, will become a linear polymer. So you can think of them all as some kind of a linear code. The basic one is, of course, the DNA, and I'm sure all of you are familiar with the structure of DNA. I just want to remind you that the code of DNA is written in atoms. Yeah. So really, if you look at the structure of the DNA, so here it is. This is double-strand DNA. It's a single strand, and as you go along the single strand, you'll see a nucleotide. Well, this is a nucleotide. This is a unit of a DNA. DNA is a polymer. This is one monomer. And if you look closer, you'll find that the monomer itself has 3 parts: phosphate, sugar, and the base. And the base is the part that is different among different letters of the genomic alphabet. So A, C, G, T are written here. Those both are atoms. Yeah, so just, I'm sure you're kind of familiar with this, but the take-home message is DNA code is written in atoms. So if you want to build a tool using, I don't know, fabrication or something that can read the sequence of the DNA directly, you know, it's kind of difficult. Yeah? This is the ultimate nanotechnology. There's nothing really conceivable that you can do to compress information even more. Like, how would you do it? You would go to subatomic particles, maybe have a quark code, I don't know. But, you know, this is it. There's no more room left at the bottom, so to say. So you zoom up into the DNA molecule, you see C pairs with G, T pairs with A. But again, the difference between C and T is really minor, so this metal group, you know, goes away and there's some other group and becomes attached. Same thing between G and A, it's just a few atoms. It's a really small difference. So DNA is negatively charged in physiological environment, and that becomes really important for the rest of my talk. Yeah, so the sequence of the DNA has been solved. I'm sure you're familiar with the Human Genome Project, which was successful. It's one of those projects, few projects that completed ahead of schedule and with a surplus of money. Can you believe it? Yeah. And I'll tell you why. But to start, let me introduce to you the sequence of DNA. Here it is. It starts with a 5 prime, and then it goes like this. Yeah. And it would take 3 million more pages just to click through them. You know, it's a really lot of information. Well, it's about a gigabyte, slightly less, right? That's a lot of information. And if you look at the sequences of individuals or different species, there are, at first look, not that many differences, right? So among unrelated individuals, we are 99.4% the same. So really 99% is the same and the rest is variance. Well, but that's still over 1 million differences, right? So we are different at the level of genetic code. So the difference between humans and chimpanzees is 99%, not so much as between unrelated humans. And again, to put it in perspective, so here's me, and here's a chimpanzee. Can you see the difference? It's right there. Very small difference, but they apparently are important, right? And of course, in addition to scientific curiosity, there are various other things where it impacts, you know. People talk about personal genomics, which is happening right now, but then why would you want to know your own genetic code? Well, because you eventually want to design drugs that work for you and not for some other person, because we're all different at the level of the genomes, at the level of expression level, so you want to tailor pharmaceutics to work for you. Another big item is cancer. You know, cancer is a disease of DNA, so to cure cancer we need to know what's going wrong, and for that we need to know what kind of mutations are there. So to develop cancer therapeutics we need to be able to sequence cancer cells repeatedly as it progresses from different tissues and so on. So it's really important to get cost of DNA sequencing down. So how do we sequence DNA, or how do we use to sequence DNA? So it used to be that number one and the only method that worked was the Sanger's method, which is basically a biology method. So what we did here is we took an enzyme that is excellent at, no, now that's too dark. Okay, put it back. We took an enzyme that is excellent at reading the sequence of DNA, called DNA polymerase, and that enzyme would read out the sequence for us. Yeah, so we didn't have the tools to build an enzyme, so we used what nature already had, this guy. So this guy can recognize the sequence of the strands that it is working on and pick the right nucleotide to incorporate it. So we tricked biology into reading the sequence for us, so, which is a great achievement, and it was an awarded Nobel Prize for it, but that was not nanotechnology by any means. And sometimes in early year 2000, there was this big push for figuring out a way to read out the sequence directly. So Illinois was a part of that. This is some early concept device, dates back to 2004, where basically a group of electrical engineers and biophysicists got together to figure out, well, maybe we can sequence DNA by pulling it through a hole in a chip. Yeah, so we have a semiconductor chip, we drill a hole, and we pull DNA through, and we read out the sequence. So kind of crazy idea, but it's actually going to work. I don't know when, but it's there. I'm going to tell you our routes to this. So again, just a brief reminder: the difference between a different nucleus is really small. You know, those are just a few atoms. So if you think, like, if you come from a fabrication background or solid-state physics background, you know, maybe it doesn't look so challenging because, you know, people have demonstrated single electron transistors and so on. So like it looked like we have a control of a matter at the level of a nanoscale. But, you know, always keep in mind that all those fantastic studies are done in cryogenic temperatures in vacuum, right? So here we want to build a room temperature device that would read out a few atom differences between the DNA molecules in water, right? So it's a technological challenge. So I like to show this slide. This slide shows the cost of sequencing DNA versus time. This is a logarithmic scale. It doesn't go very far back then. So when the Human Genome Project started in 90s, the cost of DNA sequencing was $10 per base pair. Yeah, so our genome is about 6 billion base pairs, so it's $60 billion. So it's almost mission to moon. Yeah, well, maybe not quite, but that's where it started. But luckily, the cost of sequencing DNA was dropping really rapidly. Actually, it was following the Moore's law. So that was exponential drop in the costs. But that's something interesting happened. About this time, National Institute of Health, they just drew a line here and looked when will it actually reach $1,000 or $1,000 per genome. And that was projected to be in the year 2040, which was not fast enough. And somehow they decided, well, all right, let's speed it up. So how can you do it? How can you accelerate technology development? Well, they put together a very bold program called Revolutionary Genome Technology and invited applications pretty much from all over all fields of sciences. Like there were lots of electrical engineers there. There were some completely crazy ideas put forward. Some of them even got funded. But that's not the point. You know, the point of that program was not to fulfill each of the grant to completion. It was really to find whatever works. So. And actually, because of that, it worked. It's amazing. In 2003, at this first meeting of this program, they said, well, our goal is to reduce the cost of sequencing by a factor of 100 in 5 years and another 100 or 10,000 in another 5 years, reaching goal of 2013, $1,000 genome sequencing. And that's exactly what happened. It's like one of those few examples where putting money on a problem worked. Of course, that happened not only in science lab. There were lots of different companies that emerged and busted over this time. We actually had a couple technological revolutions in the DNA sequencing landscape. So right now there are a few players still there. So Illumina is predominant platform of sequencing genomes, and you know it can be done for $1,000, maybe a couple $1,000, but it still takes quite a bit of time. But there is no limit to how fast we need to have it done. So probably in the next 10 years you'll see human genome for $100 or in 15 minutes, because the goal is that you go to doctor, they take your blood and that's it, or they swipe and you know your genome. Yeah. So how can we do it? So here's the idea that kind of kick-started the field back in the 90s. It's called DNA electrophoresis through a nanopore. So here I'm going to illustrate it using this movie. So what you have is a thin membrane placed in electrolyte solution. You have two electrodes added So you create electric field. So it's water containing salt ions. You apply electric field, salt ions will move through it. But because DNA is negatively charged, the DNA will also go through it, right? And what's amazing about it, or surprising, or fantastic, you know, after it exits, the other one will come and go through it again. So DNA usually is thought of to be like a random coil, like really messy conformations, but once it goes through the nanopore, it precedes single file, exposing its basis to this narrow constriction of the nanopore one by one. What's nice about it is that it's a self-loading system. So the physics of the system is such that the DNA finds the pore itself, enters, and then exits. Now, how do we know that it happens experimentally? So what we do, we measure ionic currents. So here's the ionic currents that you measure using in a current meter. And then this reduction is one DNA molecule, this second, this is third, and so on. So the reduction of the ionic current tells you that the DNA translocation happened. So it's really like threading, putting a thread through a needle's eye. But it's really small needle eye, and you do it a lot of time per second, per minute, 100 times. You can actually do it faster. And this happens completely without your participation, right? It just goes on automatically. So how do we use it to sequence DNA? So here's the concept that was put forward by several key figures in the field. Basically, we do this nanopore electrophoresis, but we use a really small pore. Yeah. So back in the 90s, the smallest pore that people could make was the one, again, from biology. This is a pore of a membrane channel. It has a diameter of approximately 1 nanometer, which is the size of a DNA nucleotides. So now, if we have a DNA molecule on one side and we apply electric field, so the DNA molecules will be pulled through it and exit on the other side, right? So it's just like with the solid state pore, but in this case we have a single strand DNA, and yeah. But when DNA is passing through the nanopore, the currents of ions, that go in opposite directions, will be reduced by the presence of the DNA. So here's an open pore current, and those, it'll spike down. This is one DNA molecule going through. You can zoom in. You'll see something like that. And here's a movie illustrating actually the process of ionic current happening through this pore. Ions of different kinds go up and down. But if you are lucky, or you have a rich fantasy, then maybe you can draw this trace here, yeah? And you can say, well, if we do our job correctly, maybe we can engineer a system that will be able to read the sequence of DNA directly by measuring electric currents. From a technological perspective, this is ideal because we know very well how to read currents. Yeah, we have chips in our computers and our phones that have billions of transistors. So if we can pull this off, then we can maybe sequence human genome in a massively parallel and cheap way. But that was someone's hand drawing it. You realize this is not data. So it's what we wanted to have, and that was 2001. And it used to be a fantasy until Jens Gundlach and his lab at Seattle, they actually demonstrated it. That was back in 2012. This was the first demonstration that one can actually read the DNA sequence using ionic current. So you see here there's the actual recording in time. There's clear steps that correlate with the motion of the DNA through it. So why did it work for him and didn't work for other people? Well, the key was that he used a different pore. So this pore, you can think of it, it has an ideal shape to sequence DNA. It's just, very tiny constriction, confines 3 nucleotides. And he used an enzyme here to stop the DNA motion. To basically, enzyme wants to synthesize DNA, so it pulls it up. Electric field pulls the DNA down. So it's tug of war, and the DNA proceeds through the nanopore in a controlled manner. So you can actually resolve the bases that pass. So that was really nice, and a couple of companies have formed. So one that is now a leader in the field is all called Nanopore Technologies. How many of you heard about this? Yeah, some of you have. So that's basically back like probably a couple of years ago. The prototype, it goes, it's slightly bigger than it looks. It's about the size of a palm. Yeah, but it has a nice USB interface. Here you put your sample. It has about 800 pores. So wells, yeah. So each well would have one biological pore, and it uses an enzyme to control DNA translocation through it. The accuracy of the readout depends on which hands it is. So people claim all kinds of numbers ranging from 80% to 98%. What's nice about it can read modifications of DNA directly. This is one of those, probably the only method that can read the sequence of DNA directly of the DNA, not some kind of a product after amplification and so on. And it's portable, so it's only 100 grams. You can take it in a field. So people use it to sequence Ebola when during the a couple of years ago there was an epidemic in Africa, so they took it in the field. I don't think you can take Illumina MySeq in a field. That would not work. But still, there are issues with it. So first issue is we actually don't know why it works. So it's one of those examples. We have a wonderful device. We have everything is perfect, but we didn't actually know why it worked. Let me give you an example. So here's an experiment that Jens and his lab performed. So they add a big protein to arrest the motion of the DNA through the channel, so that they can systematically determine the relation between the sequence of the DNA and the ionic current blockade. So if they take a strand made only of C nucleotides, they get current of about 40 picoamps. If they change it to T, they get 60, A about 80, and so on. So you see there are like really nice resolved levels from individual nucleotides. Well, the problem is that it's not clear why those levels should be there, because C and T are the smallest nucleotides that we have. They have only one aromatic ring, so you would naively expect them to block the current less than A and G. A and G actually blocks the current more. Well, you would think so, but they don't. So it's completely opposite to what your intuition is. And second, if you know those individual current levels, but you have a triplet of TAC inside the pore, you cannot guess what the current is. Yeah? So the currents are not additive, something funny going on. We don't know what's going on. And that's why we used our computers to figure out what gives rise to those signals. So we built all atom models of the constriction of the MSPA and simulated it, revealing in all atom details what happens there. So first of all, when you look at those movies, which are results of microdynamic simulations, you'll see that the current is not constant, right? There's a lot of fluctuations that happens at the time scale of microseconds, and those fluctuations have to do with the motion of DNA. So really, motion of DNA in the pore determines the noise level and ultimately the readout. We also found that DNA moves up and down quite a bit, and that basically contributes to the sequence flickering noise, but let's not talk about it. But the most important outcome that came out from this was the actual mechanism of why the ionic current blockades are different. It turns out that water plays a major role in determining the ionic current blockade. So if you look at this graph, and it looks like a nasty graph, but one thing you can clearly see, the black line is the current, the blue line is the amount of water molecule in the constriction. Yeah, and they have excellent correlation. And there's a very nice intuitive explanation why the current depends on the number of water molecules, because for an ion to pass, it has to exchange places with waters. Yeah? When it exchanges places with waters, then it can pass through and move on. And by analyzing results of our simulations, we could show that this exquisite sensitivity of the current comes from very complex shapes that the water takes inside this narrow constriction. So basically, if this channel was slightly bigger, it wouldn't work. If the channel was slightly smaller, the DNA would not get in. So the trick was to have the size of the channel that is just right the size for the small nucleotides to arrange perpendicular to the pore, and for the large ones not being able to do it. So that kind of gives a big signal. So that was an example of how we use microdynamic simulations to find out what's going on in a device. Now, I before said that this MinION, which is a leader in this nanopore sequencing field, has 800 parallel wells. So it's definitely better than 1. Yeah, so 800 is better than 1. But if any of you are electrical engineers, are there electrical engineers here? 1. Don't be ashamed. You know, 800 is nothing. We want 800 millions of those on a chip, right? And that's basically what we need. Well, we don't need 800 millions, but we need a couple 100,000 to sequence human genome. You know, this device is great, but it works for bacterial sequencing and viral genome sequencing because, you know, it's application limited to short, well, the DNA should be relatively short. For human genome, we need to integrate lots of them, and for that we need to go to technologies that we know how to integrate. Like, for example, silicon nanotechnology, right? So with silicon nanofabrication, we can build an array of chips, of billions of transistors, and so on. But what's nice, this is an old slide, but I like to show it, is back in 2003 from a group of Graphene. Even back then, we could make things using a nanofabrication lab, maybe even in this building, which had features comparable to the size of a DNA nucleotide. So you're looking here, this is a TEM image through a cross-section of a gate of a transistor. So you have a polysilicon, silicon dioxide, and silicon, and you have this gate dielectric layer, just 0.7 nanometers, just the size of a nucleotide. And this is a port that you can drill in such a device by TEM, and it's about the size of 2 nanometers. So even back then, we could make things that were the size of a nucleotide, so which basically stimulated development of this field. Now, how do we take solid-state nanotechnology and make a device that can read sequence of DNA? Yeah. Well, obviously, the problem with silicon nanotechnology was that all those, even how thin they were, you still end up with those polysilicon level layers that are tens of nanometers in size. So really, another key development that happened was this, the advent of graphene, right? And the graphene was thought to be ideal material for building a device for DNA sequencing because it's so thin, right? So ideally, with a graphene layer, which is just 1 carbon in thickness, you should be able to isolate individual nucleotides. And, well, maybe that's the way forward, right? So basically, the concept is the same. We have a flake of graphene on some kind of a chip. We have electrolyte solution. We apply electric field. The DNA molecule goes through, and we measure the current by we measure the DNA sequence by measuring current, right? And this is, again, a trace that my student draw. You know, this is not the measurement. So we were curious to find out what happens when DNA encounters graphene, and back in 2012, we put together this simulation. So you're looking at the DNA strand. It's already pre-threaded through graphene, and you have 2 views of the same molecule. So let's see what happens. So very rapidly, the DNA will stick to the graphene. And once it's stuck, it will keep on moving around the graphene. You can easily understand why it happens, because the surface of the graphene is so perfect. There's no preferred place for it to stick. So it's different from silica or silicon dioxide, where you have an isotropic surface. There are some hot spots where it will just get stuck. But this one, it just keeps going. And if you actually look at the way it goes through the nanopore, you'll find that the translocation proceeds in discrete steps. So look at this nucleotide. You know, it will be displaced by another one, and then another one will move in and pretty much occupy the same position as the previous one. So this is a plot of number of nucleotides versus time. So it kind of goes stepwise, which was the feature that that was nice for biological pores, where in biological pore you need to have a special enzyme to control the speed of DNA translocation. Here we can just do it with the graphene itself. So we use our methods to determine ideal thickness of the graphene that would facilitate DNA sequencing. So it turns out that the thinnest possible case, you just have 1 layer of graphene, is the worst one. because in that case you have no base ever in the DNA. So base is always stuck to the surface of the graphene, so you have only phosphate group there, and phosphate group is the same for all base pairs, so it's not going to work. So the best one in our estimates was a 3-layer graphene because it just has one base pair stuck there. All right, so we've done this, and let me illustrate another example where we can use simulations to predict something. So we had this idea, graphene is a conductor, so in principle we can charge it. And the idea was that, okay, well, maybe we can charge it and control the speed with which DNA passes through the graphene by the charge state of the graphene. So the idea was that, well, if we charge it to a negative charge, the DNA will unbind and move through the pore, and if we charge it to a positive charge, it will like hold to the DNA very strongly, it will not move, right? Make sense? And results were exactly to the opposite. So it turns out, so first of all, when we charge the DNA to minus charge, it really unbinds, but it doesn't go anywhere because it got spin here. Whereas if we charge it to a positive charge, the graphene, the DNA, it's like greasing the graphene. It just facilitates translocation because basically the DNA doesn't bind so strongly to the graphene. So that was the discoveries that we made through computer simulations, which we later implemented in this kind of stop-and-go motion of DNA through the graphene. So what you're looking at is simulations. We are trying to move the DNA strand down to the bottom, and we are allowing the graphene, the DNA strand to move because the graphene was positively charged. Now we're switching it to a negative charge, and it basically gets pinned. Now we're going to reverse the bias and move the graphene up, and move the DNA up like that, and it can't because it's still pinned by this negative charge, but we are going to change the charge to a positive value, and it just will go. And now we want to catch it before it escapes, so we will turn this charge of the graphene to a negative value again, switch the polarity of the bias, and we can do it many times. Basically, we have a kind of a way to control the DNA translocations through the pore just by changing the charge of the graphene membrane. This, by the way, also works for proteins. So protein sequencing is definitely the next frontier in this case, and I don't have time to talk about it, but conceptually you can also do the same thing with the protein sequencing. Protein would stick to the graphene, and if they're charged, you apply fields, they go stepwise. All right, this is a Plasmonics workshop, and I've been talking for 30 minutes or 40 minutes. Haven't told you anything about Plasmonics yet, right? You might be wondering why. Okay. So I realize that PlasBio, they're probably experts in this room that know more about Plasmonics than I do. So Plasmonics is definitely not what I do for a living, but I know something about it. So first of all, well, the nature of plasmonic resonances, right? You have metal particles, you apply E field, and then those electrons would oscillate back and forward, and if everything is done right, we'll produce a standing wave. All right. So it has been shown experimentally that this standing wave resonances can be used to trap proteins, right? So this is work from Erwin Gordon and Victoria. So it's one of two of his papers. This one where he demonstrated trapping of a single protein. So what you're looking at is, I think it's called a keyhole assembly. So this is metallic layer. You apply E field with laser, and then there's this standing wave here. And the proteins are in solution. They come in. They block the optical transparency. This is not electric current, by the way. This is not electric. This is just optical transmission. And you can kind of see several levels. And you have, if you have fantasy, you can say maybe protein unfolds here. Yeah. But definitely, you have these two states: protein folding, unfolding, vacant. But you can trap molecules there for some time with that. So the other piece of information I need to remind you, this surface enhanced Raman scatterings. Again, you're probably experts here. So for those who are not, basically, generally when a wave hits a molecule, it excites it, and then the excitations come back to the same state and lets you have a normal scattering. But sometimes, you know, it goes to a different state, which is a signature, a chemical signature of molecules, a so-called Raman shift. And that's been known for some time. And this used to be a really, really weak process, right? It's something that it's difficult to measure unless you have some kind of a surface roughening. So this is an example of a measurements where you have a rough metallic surface that enhances emissions. So in this case, the enhancement was a factor of 100. But generally, the idea is that this Raman, the probability of seeing a Raman event goes as a force power of field vector or second power of field intensity, right? So that's why this kind of, you have a dramatic enhancement in Raman cross sections when you have a rough metallic surfaces. All right, so that you're probably familiar with. Now, this piece of information is Raman spectra of different bases. So you're looking at G, A, T, and C. And this is so-called Raman shift. So that's basically by how much this relaxation is off from its input value. And if you look closely, you know, all of them have different signatures. Yeah, so that's great. Although I must say this is not single molecule, right? This, when it was done, it was, I think, this particular paper reports from a bulk measurement. All right, and so in principle, you know, Raman can be used for DNA sequencing in principle. And final information is this paper was published in 2013. I believe that was the first one to demonstrate, you know, a combination of a nanopore with a Raman detection of nanoparticle transport. So in this particular measurements, which was by Joshua et al. And Tim Unblack from Imperial College in London. So what they did, they took a nanoparticle and they coated with lots of dyes. So it's not a single molecule per se. They don't detect a single molecule. There are like maybe a couple hundreds of them. But they can simultaneously measure the current blockade by this particle and the Raman emission, you know, that shows the all those things were together. When I went to Delft, Delft University in the Netherlands, that's where I learned actually the plasmonic nanopores. Okay, so how do we make a plasmonic nanopore out of a normal nanopore? So very simple. We add two old bow tie triangles, and then the idea is the following. Again, it with laser light, and if we tune our laser properties correctly, we'll create a pore. So good. All right. And here's basically from that concept for using Raman. All plasmonic DNA saying the idea is kind of complicated, but probably for you it's not all complicated. Basically, we have a nanopore there. We have a DNA molecule that wants to go through the nanopore because we have applied electric field. Okay. Now we also have this laser light that comes in and generates a standing wave, which will trap the DNA directly, and it will also stimulate Raman emission from the hotspot over here, where the DNA is. And we will look at it at 4 different frequencies, and kind of by measuring the Raman intensity, we hope to decipher the sequence of the DNA that passes through. You know, this is concept, right? This is just a picture. So I'm going to tell you how far we got with it so far. This is work in progress, okay? But before I go that, well, let's just, why this is, if it works, why this is great? Well, because it's a direct measurement of chemical information. So we say if we make it work for DNA, we'll make it work for RNA, we'll make it work for protein. You know, if it works, it's a big deal. Although it's difficult to pull it off, as you will see, but it's potentially revolutionary, okay? The other thing is this plasmonic tweezing is a method, it's basically a different physical there's different kind of interactions to control DNA speed, right? So one question that I frequently get, well, why do you need to bother with this plasmonics? Can you just reduce the electric fields that drives the DNA through the pore? Well, no, it doesn't work because you have all these capacitances. You cannot reduce it fast enough to advance the DNA by one base pair. You cannot. But with plasmonics you can, because you can switch off the laser light, within nanoseconds. There's no problem. There's no capacitance in optical sense. Yeah, so that's why we actually went for this project. But of course the question is, can it be done? And I'll tell you how far we got with it. So the first question is, well, can you actually make something like that? Could you turn off the light at this point? Can you actually make something like that? Like, is it at all a structure that is possible to make? The answer is yes. Here it is. You have two bow ties. And this is from his writer's lab at Netherlands. But you realize this is not every person can do it, right? So it's equipments, and it can be done. So we can make more. We can make another pore. There's just one. There's more than one. Now, does the system actually work? Let's do anything plasmonics. That's the question you probably ask. And that's again a paper that Kees Dekker's lab published. So what they did, they skimmed the surface. They made this device. They skimmed the surface, and they see how the ionic current depends on the position of the focus along the membrane. And they think, when you have a high increase in ionic current, that means you locally heat the structure. I assume everyone knows that this plasmonic resonance, you have local increase in temperature. And far away it doesn't matter, but that's basically the way to localize the pore. And you can kind of reconstruct the actual profile of intensity. Because you know how the ionic current depends on the temperature. So you can do a separate calibration measurement for temperature dependence of ionic current, and then you can use this simulation to reconstruct the actual shape of the optical beam. All right, so this kind of works. Now, can we do nanopore translocation experiments with it? So this is, again, a paper from Dekker's lab. So what you're looking at are DNA translocation measurements done at different intensity of the laser. So this is conventional translocation. You see there's no DNA, there are no downward spikes. When you add DNA, you start seeing those blockades. Yeah, everything's usual. When you add the switch on the lasers, the first thing that you see is the current baseline increases, just because you hit the solution. But you still get those blockades, yeah. So you still get the DNA going through, so that's great. Now, so that was good, but fabricating those structures is challenging. Okay, let's not be shy about it. And if you want to build a device, maybe that was the first thing that people ask: how are you going to make 1 million pores like that? Well, it turns out that there is actually very neat trick that you can do to make those pores without really using a TEM. So it's called so-called self-aligned nanopores. So here's the one that is fabricated using TEM. And here's one that is fabricated using so-called dielectric breakdown method. So dielectric breakdown method came from the lab of Vincent Tabartkosa. He's in Ottawa. So the idea is we have the membrane, it had no pore in it, right? Just a thin membrane, no pore. We apply very strong electric field. We're talking about 10 volts. Eventually the membrane will break. And when it will break, it will create a pore. So the idea is the following. So you apply electric field, and you wait for the leakage current to rapidly increase, and once it increases, you switch off the field, right? So you make the pore by dielectric breakdown, and that's what this Canadian group published. What Delft people did, they discovered that if you have a plasmonic structure that is active, meaning you have plasmonic resonances, then as a result of this breakdown, you will produce the pore exactly in between of the two bow ties. Yeah? So that's kind of an important advancement. So you don't have to find what the TM, what those things are. You just apply the dielectric breakdown, and it just appears right in the middle. Yeah? And experiment has shown that those pores are as good as the ones that you make with TM. All right, so how do we model those systems? And that's basically what Chenyu's tutorial will be about. He will give you a really simplified version of the simulations that I'm going to show you. Yeah, and the way we did those simplifications is that you could finish it in the afternoon. Okay. So the simplifications are that you don't have water molecules, and system is slightly different, but basically everything that I'm showing you today, if you go back and you want to reproduce it using our tutorial, you should be able to do it. So here's a simulation that illustrates motion of DNA in electric field. So in this case, we have no plasmonic resonances, nothing. So just a DNA molecule, but we have those corn-like structures that are the gold bow ties. So DNA goes through. So the motion of the DNA is a little bit funny because it sticks to the surface of the silica pores. But anyway, so it goes through, and the motion, the velocity of the DNA is proportional to the field that we apply as expected. All right. So now we need to build in somehow the plasmonics in our molecular dynamics simulation. It hasn't been done before. I believe you were the first one to do it. So if it's not right, please let me know. I'll be happy to acknowledge other people who've done it. But the basic idea is that we have our optics simulations, like in your case would be DD-SCUT. So back then when we did this study, we used some other software. But it's basically the same. You solve optics equation, you get this field profile. And then what we do is we modified our molecular dynamics codes so that we can add this plasmonics field on top of our molecular dynamics simulations as an external potential. And then we assume that the local force of that plasmonic field is proportional to the gradient of the field times some dielectric mismatch between the molecule and the solvent. So that's an assumption that we make. So I realize that there are more sophisticated way of doing it. But as a first proof of principle simulation, that's what we did. So here's like a cross section showing the actual field lines. That I believe those forces show local forces on a single nucleotide that would be at the edges of those structures. So let's see how the simulation works. So here's one simulation, one of the first that we've made, where we had this plasmonic field on. Those semi-transparent surfaces indicate the presence of a plasmonic field, and those errors indicate the local plasmonic forces applied to individual nucleotides. Okay. So in this case, the DNA is trapped by a single hotspot, and we can repeat this simulation. So we always have the same driving bias. The only thing we change the laser power. So if we increase laser power above a certain threshold, we apply enough field to deform DNA and make this kind of a knot here. So DNA is trapped by two hotspots. Some plots illustrating the effect of plasmonic field on the DNA translocation. So there's no field, it goes like that, and then as we increase the field, eventually it becomes trapped. So that's good. We, of course, with the simulations, we're limited in time scale, but we've shown like factor of 100 reduction of DNA translocation velocity. So the next thing we explored was what happens if we try to step DNA through this plasmonic nanostructure by switching the laser beam on and off. So that was basically one of the major ideas here, that we can switch off the laser like every nanosecond and see what will happen. So here's a simulation that illustrates what happens. So we have DNA in this double hotspot trapped. We switch it off, the DNA will move down, and then we switch the plasmonic field back, and it will get trapped again. And we can repeat it as many times as we have DNA left. In this case, we repeat it, I think, 6 times. and so on. So at the end of the day, we can obtain a plot of DNA transport versus time. So you see, we were able to obtain this nice stepwise. I mean, they're not ideal by any means, but this nice stepwise translocations, which we were looking for in the case of biological pores and also pores that were having graphene membranes. But still, those are not single base steps. So the steps here are about one turn of a helix. We actually get much better results when we consider a system where there is only one hotspot. So experimentally, you would do it by designing an asymmetric bow tie. There are quite a few ways you can do it. So with a single hotspot trapping, it actually works. We could achieve much smaller steps. So continuous trapping, DNA doesn't move. And then, so what we can do, we can design different pulses of the laser beam, and eventually we can get something like about, on average, one base pair. But again, this is like an experiment. This is not ideal. Yeah, you get what you get, and I think it was our best result. On average, one base pair per step. So here's an illustration. We can also do what we can do. We can now take this ensemble of microscopic information and predict what would be the actual Raman readout. And the way we do is the following. We assume that this emission is independent of individual bases. So for each base, we know the local electric field. We know local electric field, and we assume and we know the enhancement factor for Raman emission, and we just construct a probability. So if this is the DNA molecule going through the pore, so we can basically predict. So this is plasmonic field is off. Now once it's on again, we can tell in which, what would be the Raman shift signatures from this kind of simulation. So there is a way to take a microscopic information, do the trapping, and then actually transform into what kind of source signatures you'll see from it. So here's an example that we did. We looked at first at the static information. So we have only one DNA molecule, so it's trapped. And to simplify things, we decided to do the following. We take a piece of double-strand DNA, which is entirely CG, and then we add a block of 18 nucleotides. And then we consider a process where we kind of shift this 80 block along the molecule. Okay, so that's basically what we have. And then you can look at this intensity, Raman intensity, and you can see that so this is a CG channel, so intensity drops because 80 blocks comes in, and then intensity in the 80 channel increases, right? So that's something you would expect. Then we can do the same for smaller blocks. So if we have a block of just 5 or 6 80, we actually see 2 spikes because the DNA goes through 2 plasmonic hotspots. Finally, we can look at individual base pair passing through it. And so you'll get 2 spots, but you will see that they're actually not the same height. And this has to do with extreme sensitivity of the emission to position of the base pair. So actually TA and AT will give you different signatures if they're placed in the same spot, just because they're different distance away from the hotspot. So what about signatures that we expect to record from the actual translocation event? So here's a simulation where we have the single hotspot trapping. We turn the field on and off, and you see that some of the nucleotides light up. So those are the nucleotides that emit a majority of the intensity in a given spectrum. So the nice thing that we found is that if you look at an individual channel, more than 50% of the intensity is determined just by the 3 bases that are in hotspot. I mean, ideally we would want to have 1, but it's impossible. So we will have some sort of a deconvolution problem when we want to read back the sequence of the DNA. But the good thing is that we have 4 channels to work with. With ionic current, we usually have only 1. So yeah, so that's where we are. So if you look at the pattern of these hotspots from the DNA, so we kind of cover the entire DNA strand as it passes through. But of course, we have emission from both strands of the DNA, and we have quite a few nucleotides emitting. And it's actually not linear. If you look closely, sometimes you have, the highest intensity spot would kind of jump back. It just depends on the pitch of the DNA. So the convolution problem here is quite major. So that's why we thought, well, okay, so that was nice. So we published this paper, but then, so what happens if you do it with a single-strand DNA, right? So single-strand DNA should be much easier to work with. So one thing that I don't have a movie for, but you can easily imagine, if you take a single-strand DNA and you try to trap it with a plasmonic field, you'll get a coil, okay? Because single-strand DNA is a flexible molecule, it will get attracted and will just form a blob here. You will not have a strand threading there, you will not have a strand threading there, right? So, and that's, I believe, Chinyu, that's what you see, right, in your trapping simulations in the tutorial. So you will actually do this, but you'll do it with a piece of protein. Okay, so you'll just have a piece of protein, you'll just trap it with a plasmonic hotspot. And you'll see like a coil. But a year ago we decided, well, okay, let's just make things easier. Let's just apply tension to the other strand. So basically what we did, we apply tension to the other strand to pull it away from the plasmonic hotspot, and we have electric field that pulls it in, and this guy kind of traps in the middle. Make sense? And then everything kind of goes, concerted like this in this process. And we are looking at, again, the source emission in 4 different channels corresponding to different nucleotides. And well, in this particular simulation, you can kind of count the number of peaks, although this is kind of difficult to resolve. But basically, you can see 9 peaks corresponding to 9 nucleotides in this kind of simulation. So that's kind of ideal scenario. It probably won't be any better than that. So that was nice. So we published this paper, and then the question is, well, but how can we actually do it, right? How can we do something like that? And here you go. So the question is, well, let's use 2 pores. 2 is better than 1. So if we have 2 pores, and we trap the DNA molecule with 2 pores, then basically the DNA will be subject to 2 forces pulling it down, and that will create the tension, right? So that was the idea, and very happily we have this paper published yesterday. So it's available and just accepted in our letters. And of course the idea is that now, we'll do it like that. Have a bow tie, we have a pore through one, and we have another pore nearby. And maybe we can control the velocity by plasmonic or other means. But just to show that this is not science fiction kind of things, this is actual device. So you see there's a pore here. There's another pore. Yeah, and in this paper we describe, there is no plasmonic there, but we describe the method of actually trapping the DNA. Yeah, it's basically proof of principle that with 2 pores we can trap the same DNA molecules. It actually looks like that. And that kind of dramatically increases the residence type of the molecules in the pore membrane. So that's pretty much all I wanted to tell you. So let me finish up with acknowledgements. So this project is funded by NIH. Also, we received funding from Oxford Nanotechnology. So take all my comments about Oxford Nanopore with that in mind. A computer time comes from University of Illinois and Exceed. Those are people from my lab who participated in the project that I showed you, and those are my great collaborators from TU Delft, and thank you very much for your attention.